So hi, I am. We are starting to start a new chapter. That is chapter 4, and the chapter 4 deals with genetics. And genetics. The word genetics, of course, you can understand that it has to do with genes, but it's mainly related to inheritance of characters from one generation to the next. So why it is interesting, because first there are many diseases. There are many diseases that is related to inheritance. For example, color blindness, color blindness, and it could also be related to, for example, cystic fibrosis. So there are many diseases which could be passed on from one generation to the next generation. So it is very important to understand the mechanism of transmission of diseases. Now, the central to the transmission of genetic disease of a genetic disease is chromosomes. And why chromosomes? Because chromosomes are made up of DNA plus proteins, out of which DNA carries the element of being passed on from one generation to the next. That is called as character. Now, the most important, to start with, the most important aspect of genetics that I would share is the starting point of genetics. The starting point of genetics is Gregor Mendel, who is now called as father of genetics. Now, try to understand that why he's called as a father of genetics, although his research was on pea plants, and he was not a biologist. He was a naturalist. He was a monk in an Austrian church. Gardening, he used to garden. He used to do a lot of gardening, and he was a very sharp observer of plants, and he would try to understand why plants look the way they look, because he noticed a lot of variations in the plant, and he wanted to understand that what is the basis for this variations. For example, he would be puzzled if he would see when tall plants when he would grow pea seed, he will see tall and short plants. So what's the basis for tallness and shortness? He is putting the seeds which are very similar. Why some flowers are purple and why some are white? Why it is not a mixture of white and pink, white and purple, like pinkish color? So why these characters, they vary, when the starting point, the seed is same, why there is variation. So that was his, question that he used to think a lot about while gardening. And since he was a very good mathematician, he was very good at maths, so he could he tried to understand and try to give logic to his observations. And his observations came to be known as Mendel's law of inheritance. Now, the question is why he is called as father? It is father because through the inheritance of characters in pea plants, we can understand inheritance of many human diseases. So, for example, color blindness, the passing of characters from one generation to the next generation in peas could explain the passing of some disease characters from one generation to another generation in humans. So collectively, because of his work, which was in somewhere in mid 18th century, his work is still very valid, and so he's called as father of genetics. So let's see how this chapter starts. This is chapter number 4. And what are the learning objectives of this chapter? Is that understand the difference between genotype and phenotype, understand how to use the principle of genetics to predict genotypes and phenotypes, difference between dominant and recessive, difference between dominant and incomplete dominance. So compare and contrast different kinds of sex-linked diseases. So before we go into this chapter, it is very important to understand few definitions. And they're very short, definitions, but it is very important to understand these clearly before we move into the chapter. The first definition is that I want you to know is phenotype. So phenotype refers to physical attributes. In case of plants, it could be tallness or short in their heights, or the color of the flower. In humans, it could be color of the eyes, color of the hair, again, height. So all these, so phenotype refers to phenotype refers to physical attributes that could be watched just from the naked eye. And this would be, for example, height, color, pattern of leaves, so on and so forth. So physical, that can be seen direct from the eyes. It won't need a microscope, it won't need a magnifying glass, it won't need DNA. The word genotype refers to genetic constitution, which means that every character that you see, every character that you see here is coded by a gene. And genes are located on the chromosomes. So if you are seeing that somebody is tall or short, it is because of direct related to genetic function, the functions of the gene. Okay, so that's genotype. So for example, let's say purple, a flower that has a phenotype of purple color, it is denoted by big B and big B. A flower that is purple, it is denoted by big B and small b. So I would like to focus your attention here. Big B and big B, they are same genes. They are identical. So they are called as homozygous dominant. So it makes perfect correlation that two big Bs, when they come together, two chromosomes, one from father, one from mother, or one from male plant or another from female, when they come together, big B, they become homozygous because they're identical and the color is purple. This is an interesting situation where one of the chromosome has big B and the other chromosome is small b. So they are different. Despite both are responsible for color, but the genetic elements are different. Big B, small b. This is called as heterozygous, alternative characters. Alternative characters. So interesting thing is that in this heterozygous condition, still flower is purple. Right? Now I'll try to explain more. In this small b, when the two chromosomes containing the genes for colors is small b, homozygous, they're identical, it is white. Now, why do we use the word dominant and recessive in genetics? So when big B comes, right? So it becomes purple. It's clear. When small b comes, it is white. It is also clear. But when there is an alternative characters, big B and small b, so you would see that if one would expect that it should be 50-50. So this should be pink or some other color. But when you see this is there is half of big B, half of small b, or 50-50 contribution in this character, but still the character is purple, which means that the purple color is dominating. So a character that dominates over the other is called as dominant. So homozygous character, homozygous when two chromosomes, two genes are identical, and that character expresses more in a situation. That's a dominating character. So we covered phenotype, genotype, dominant, the character that expresses itself, recessive, that is hidden. Now, I would like to emphasize here that in this heterozygous condition, which has big B and a small b, heterozygous condition, which has big B and small b, so still the flower is purple. So the white color is not there anymore. But don't think that the white color is lost or deleted from the chromosome. It is hidden. It is masked by the purple color. So remember this. Recessive means suppressed, not removed, not deleted. Okay. Some of the characters that are dominant in humans, for example, tongue rolling, ear lobe attachment, or arm folding. So these are some of the characters in humans, also like plants, which are dominant versus recessive. In plants, the height of the flower is the tallness is dominant. The color of the flower, purple, is dominant. The color of the seed, purple, is dominant. Round colored seed is dominant. So all these characters which are on the left side, they are dominant. Okay. Now, as I mentioned to you, genes are located on chromosomes, and they are located on chromatids. So for example, growth genes is located here. So genes are located at a specific position on a chromosome, and that is called as loci. So the gene for hair color is here. The blood type and eye color, they are on the same chromatids, but they're at a distance. So location of genes is called as loci. Now you will hear in the world of genetics a word called as alleles. So when a single character, so for example, what do I mean? That a single character, so let's say that, let's consider that character as color of the flower of pea plant. Now, if this color is purple, so it could be denoted by P. And since a pea flower is a diploid part of the plant, so it will be denoted by big P, big P. A flower could also be purple if there is big P and a small p. Big P coming from one of the, let's say from a male plant, and small p from the female part of the plant. So this is, because the purple color is dominant, this is called as homozygous dominant. This is homozygous recessive. Oh, I'm sorry, this is heterozygous recessive. Homozygous recessive would be this. So what you see, these two are alleles. So alleles are same here, big P, big P. Here, big P and small p, they are different. Why different? Because small p is for white color flower and big P is for purple color flower. So color is one of the characters, and this character can be represented by two alleles. These two alleles, if they are same, identical, it is homozygous allele. If the alleles are different, it is heterozygous. Again, if the alleles are same, again it is homozygous. Now because purple color is dominant, so this becomes homozygous dominant. This becomes heterozygous, and this is homozygous recessive. The white color, so purple color is homozygous dominant, right? This is heterozygous, and this is homozygous recessive. So we have to use these terms, phenotype, recessive, genotype, dominant, homozygous, heterozygous. So heterozygous is alternative character, big P and small p. Okay. So this is a brief introduction to genetics. So here, this allele is represented by two genes, big A and small a. So if two big A's are present, the phenotype could be different. If big A and small a are present, it could be different, or it could be same, depending upon whether it is homozygous or dominant or homozygous recessive. So here if you see big B and big B, this is homozygous. Big B and small b, heterozygous. And small b, homozygous. Now, if big B is dominating over small b, then big B becomes a dominating character, okay? So with this background, we go into learning about genetics. So let me give you some more, a little bit more concept. And the concept I'm giving you is that, let's say a colorful flower, purple, is denoted by P. Now flower is a diploid, so it will have two sets of a pair of genes. Now, this is diploid two cell. Now we know that flowers have reproductive organs, male pollen and female ovary. So these reproductive part of the flower, they will undergo meiosis to produce gametes. And as you remember, production of gametes from your class, last class, chapter 2, is through a process called as meiosis. So this is 2n is referred as diploid, and 1n is referred as haploid. And the fusion, the fertilization, always takes place between two haploid gametes. It could be like this, or it could be so. Now, what is small p? Now, big P refers to diploid character, diploid, and refers to purple. And small p, so big P refers to, as I mentioned earlier, purple, and small p refers to white color, right? And we know that purple color is dominant. So let's go to the next concept. So when they undergo meiosis, they produce haploid gametes, and they produce haploid gametes. Now, what Mendel did, he crossed the reproductive parts, and then one would expect that the next generation would be big P and small p. Now here, Mendel expected that this one should be either 50% white or 50% purple or in between, but he got very alarmed because this one, heterozygous, 100% of them were purple, right? So he got confused that maybe something is there. So he did this experiment many times. So again, I said that he was a mathematician, statistician, so he started looking at pattern. And when he got a pattern, he thought of putting it as a experiment, as his hypothesis. So, so let's look at this slide. So what you see here, a purple flower and a white flower. So this is appearance. Appearance means phenotype, the colors. And what is the genotype? What is the genetic makeup? So big P, because this is diploid. Small p, that stands for white flower, diploid. Then if it has to reproduce, fertilize, then it has to be done between the gametes. And gametes are formed because of meiosis. So what you see here is that formation of a gamete. So this is the formation of a gametes because of meiosis. Now Mendel crossed this. Cross means took male gametes and the female gametes, and he crossed. And this he called as F1 generation. This he called as F1 generation. So in this F1 generation, he thought that because there are two different kinds of phenotype, he would expect this to be in between. Just like if you mix two color in a glass, So one would expect an intermediate color, but he got 100% of the purple. This happened because purple color is dominating. But Mendel was very concerned why this was happening. So he repeated this experiment many times, and then he wanted to explain what was going here. So he created a statistical square. This statistical square is called as Punnett square. So he used Punnett square to explain what was going on. So what is a Punnett square? So a Punnett square could be I'm just drawing here. It could look like would look like this, and here on the right hand side or left hand side, one can take female gametes, and on the top one could take male gametes. So a female gamete would be on this side, and the male gametes would be on this side, right? So he was confused that why, he always wanted to ask why this situation is leading always to purple. What is happening to the white color? That was his biggest dilemma. So what he did, he came to a situation where he thought that this is the alleles which he has to work on. So what will happen if this undergo meiosis, right? So he realized that this will produce two kinds of gametes. One gamete would look big P, and another gamete was small p, right? So this is one situation. So a heterozygous, why this is heterozygous? Because big P and small p. Homozygous, whether it is big P or small p, they will always produce one type of gametes, only big P, or only one kind of gametes, which will be small p. But a heterozygous will produce, heterozygous will produce two types of gametes, whereas a homozygous will produce only one kind of gametes. So what Mendel did, female, he had big P and small p. In males again, he had big P and small p. So now what he did, he crossed this with this with this, and he crossed this with this, and then this with this. So what did he get? Here he got big P. Here he got big P, small p. Right? Now when he crossed big P and small p, he again got big P and small p. And most interesting, when he got the small p here, then, so this big P was purple flower. big P was purple. This flower was purple, big P. This was a purple color flower. Big P and small p, they were also purple color flower. This one, this flower, as well as this flower, they were purple color flower. Why? Because big P is dominating and big P stands for. Only interesting aspect here was that when small p comes together, then the flower becomes white, which means that small p becomes a recessive character. So let's go back to the slide. What you see here, so you see here three purple colors, big P, and small p. So three purple colors. So by phenotype, by eyes, by physical attributes, there are three purple and one white. So the phenotypic ratio here is 3:1. But when you look at genotype here, one big P, so that's one, two big P, two big P and one small p, that is heterozygous. So 1:2 and only one white. So the ratio of genotype is 1:2:1. So to make the long story short, when Mendel crossed purple and white flowers, he got phenotype ratio as 3:1. He got phenotype ratio as 3:1, whereas he got genotype ratio as 1, which was big P, 2 as big P, small p, and 1 as small p, 1 as small p. So this is the outcome of one of the experiment that did. And because one of the character is dominating, Mendel called this hypothesis in his time as law of dominance. Okay. So now with every law, there are some exceptions. So when a work is done, every scientist try to repeat that kind of work and see whether that phenomena holds true for other situations. So one of the scientists, he tried in this flower, which was red and white. So he denoted the wild type by big C, and the flower was red color. So superscript R and R, and denoted the white color flower with CW. When this flower under the reproductive organs created gametes, they underwent meiosis. So the red color flower generated R gamete and W. So this is after meiosis. In F1 generation, you get to see that, very interesting, unlike the law of dominance, you see that when these two flowers mate, the F1 generation, F1 stands for first flyal 1 generation, or first flyal generation, as pink, which means that these two colors blended with each other. And the result flower was, in F1 generation, was pink. So it was not the case of dominance. It was not complete dominance. So this was called as incomplete dominance. And when they did the F2 generation cross, they found that the phenotype was one red, two pink, one flower. So what was the phenotype here? The phenotype is 1 to 2 to 1. And the genotype is again 1 to 2 to 1. So in case of this plant, or let's say in case of incomplete dominance, in case of incomplete dominance, the phenotypic ratio is 1 to 2, 1 to 2 to 1, and the genotypic ratio is also 1 to 2 to 1, which is in contrast to the complete dominance, where the phenotypic ratio is 3 to 1 and incomplete is 1 to 1. So now I mentioned to you that Mendel is still called as father of genetics because his work could be extrapolated, could be extended to color blindness. So color blindness, the story of color blindness is that one should understand a very interesting phenomenon that happens in humans. Is that in humans, the sex chromosome that distinguishes male and female is a 23rd chromosome. 23 number chromosome is referred as a sex chromosome. Why? Because in males, this 23rd chromosome is X and Y. And in human females, this chromosome, sex chromosome or 23rd chromosome is X and X. So pay very important, pay here attention that pay attention here that if you see, focus on male 23rd chromosome, one is X and the other is Y. This X and Y, they are very different from each other. In contrast to the females. So what do you have in females? In females, this both are same. This both are same. The both sex chromosomes are same. So what is the consequence that males have different X and Y and female? What is the consequence? So the consequence comes in context of, so let's say that there is a problem. There is a problem with a male. Then, and this problem will occur on X chromosome. So this is a problematic X chromosome. So this male will always exhibit disease. Always exhibit a disease. Why? Because there's only one X chromosome, and if it is bad, then there will be disease. In contrast, females, if one X chromosome is bad, then it will be rescued by the other chromosome. So in males, if one X chromosome is mutated or there is a problem, then 100% chance that male will exhibit disease. But in females, if one X chromosome is bad, that bad chromosome will be rescued by the normal chromosome. This phenomena in females is called as dosage compensation. So if X or Y, if it is damaged in male, males will always show the disease. But in female, if only one X chromosome is problematic, then the second chromosome, X chromosome, which is normal, will rescue the diseased chromosome. Unless both the X chromosomes are bad in females, if the two chromosomes, both X and X, are bad in females, then only a female will exhibit disease. So this is the case that we see in color blindness. So you see here in this case, so what we see here is that so a normal mother, this mother is normal despite there is a problem with one of the X chromosome. So mother has a problem, but she is normal because her one of the X chromosome is normal. When she gets married to a normal father XY chromosome is totally good. So what happens when they get married? When they get married, the girl is, if they have a girl, they will be normal. If one of the X chromosome is bad, then again, this girl child will be a carrier of the disease. But look here, very interesting. The male child, X chromosome is damaged. So this male child is colorblind. So if one X chromosome, so now how this male child got this chromosome? This is because of crossing over. Remember, meiosis 1, prophase 1, crossing over occurs. The gene from the mother went on to the father because of crossing over. And this child is male child and he is colorblind. His X chromosome, because he has only one X chromosome and that is damaged, so he has to have the disease. Let's look at this phenomenon here, that when a father who is colorblind gets married to a normal mom, so the daughter is normal despite being carrier, and the son is totally normal. So there are various permutation and combinations. So this chapter is just to introduce to you genetics. Genetics is inheritance of characters from one generation to the next generation. This field, this area of biology was developed, initiated by Gregor Mendel, who's today also known as father of genetics, because his work on P could be related to, we can understand human diseases by learning his work. So he's the father of genetics. So with this, we finish chapter number 4.